Hi everyone, my name is Shresta Antil and I am from Boston Ridge, New Jersey. My topic for my research paper is bispecific RNA aptamers for mediating blood-brain barrier translocation via insulin receptors and precision targeting of EGFR variation 3 glioblastoma cells. So to start off, what is glioblastoma? It is an extremely aggressive and deadly brain cancer that has an average survival rate of just 14 months. Current treatments, such as surgery and chemotherapy, have severe side effects and also risk a chance of recurrence. Symptoms can begin with pounding headaches, but quickly evolve into damaging seizures, paralysis, vision problems, and disorientation. This cancer is also incredibly rare, with only 12,000 new cases every year, but over 200,000 dying annually. Out of all of the cancers, glioblastoma has the lowest 5-year survival rate as well, at less than 5%. The causes are relatively similar to that of other cancers, whether this be environmental factors such as radiation, being at a greater age, around 70, or a family history involving DNA mutations. Unfortunately, it is a highly invasive brain cancer that disrupts healthy neural tissue, and it leads to severe symptoms such as headaches, seizures, and cognitive impairment. But the biggest challenge in treating glioblastoma is the blood-brain barrier, or the BBB. And basically, what this is a selective membrane that prevents most drugs from even being able to reach the brain. Given these circumstances and the impact that glioblastoma has, we urgently need new therapeutic strategies. My research focuses on a strategy utilizing bispecific aptamers, which are small single-stranded nucleic acids. Unlike antibodies, aptamers are much more stable, easier to synthesize, and can be modified for enhanced specificity, much easier to work with. The small size of these aptamers will allow them to cross the blood-brain barrier by binding with the insulin receptors that are on the membrane. As displayed in the image, the aptamer can initially be carried via a nanovesicle to reach the membrane. From there, it'll bind to the insulin receptor, enter the brain, and target the cancer cells that have the mutated receptor. But through computational modeling and molecular docking simulations, I aim to design and validate an aptamer with high specificity and affinity for the insulin receptor EGFR and EGFR variation 3 to potentially revolutionize cancer treatment. I also hypothesize that these aptamers will bind to the specific binding sites of the insulin receptor and the EGFR variation 3 to aid drug transportation in crossing the blood-brain barrier and to target the cancer receptors. To develop an effective bispecific aptamer, I use several computational techniques, which are broken down into the following steps. The first step is receptor modeling, which is specifically focused on the insulin receptor found on the blood-brain barrier, the EGFR receptor, which is on healthy brain cells, and the EGFR variation 3, which is on our glioblastoma cancer cells. This process will begin using UniProt, which is a database that has information on various protein structures and functions. This provides access to the amino acid sequences for all of our receptors. From there, I used AlphaFold 3, an AI-based system which can then generate image predictions of the protein's physical structures from their sequences. This step is really important in visualizing and actually building my model for the receptors. Finally, through the deep learning-based network ScanNet, I predicted the locations of all of the binding sites via binding site analysis for the three receptors. Next, I focused my attention on the aptamers and constructing their models. The first step is to utilize the unifold server, which takes an RNA sequence to create an image of the structure. This system provides a visual representation of how the amino acids interact, and it'll download as a CT file. I then uploaded this file to the CT web server, and it provided me with a dot and bracket notation for each aptamer. With this notation, I then used a server called Rosie, and Rosie takes the RNA sequence and the dot and bracket notation to queue a job and provide me with 3D images of my aptamers. Finally, we can move on to molecular docking simulations. Now, this step involves the server HDoc, which can predict and visualize binding interactions. It'll provide several different situations based on the molecule structure. So red zones will highlight optimal binding sites, and then blue or white areas are areas that are less likely to be binded to. Then I actually simulated the docking and interactions between the aptamers and the receptors to figure out which aptamer would be most successful. In order to ensure that these trials were accurate, I used PLIP to finalize my analysis and put together final details. Now, PLIP is a tool that identifies molecular interactions through hydrogen bonds and hydrophobic contacts. So this tells me more about the aptamer's stability and specificity when it binds to the receptors. Lastly, I compiled all of this information together through an app called Chimera, which helped me put together images and to clearly represent my analysis, which is how I am able to show it to you today. After the computational steps, I identified a few key findings. These diagrams are my results from my initial receptor modeling. So the image shows the binding sites of each receptor and where to keep an eye out when it comes to the aptamer interactions. If the aptamer is binding at the red sites, that means that it's a strong bind. But if it's not, that means that the binding is poor and much more weak. Here, we see my results from the aptamer modeling. Now, this is the table of the sequences and the dot and bracket notations, which I had then later uploaded into Rosie, which had queued the job. These are the 2D representations of the 4 aptamers that I analyzed throughout my research. As a note, the 3D files were used in the actual simulations by being folded in the manner displayed in the 2D representations. So, what did I determine? So through my modeling and my predictions, I identified that aptamer 1 demonstrates the best binding interactions among all 3 receptors. This is because it is able to successfully bind to the insulin receptor. That means it'll be able to cross the blood-brain barrier, as compared to another aptamer, for example, aptamer 3, which doesn't bind at a primary site, showing that it wouldn't be able to even enter the brain. Additionally, aptamer 1 showed successful signs of binding to the EGFR variation 3 receptor, but not the EGFR receptor. This is really important because it means that aptamer 1 is one of the only that can avoid binding to the healthy cells, but instead target the cancer cells at a much better rate, reducing potential side effects on healthy brain tissue. Aptamer 1 also proved to have strong binding affinity, which is the strength of the bonds. The docking simulations revealed that there are greater hydrogen bonds and salt bridges between aptamer 1 and the insulin receptor. This shows that there are more interactions, therefore stronger binding, as compared to the other aptamers, which are much lower in the graph. Similarly, aptamer 1 has lower binding with the normal healthy receptor, but successful binding with the glioblastoma receptor. Now, this is a more quantified way of demonstrating how aptamer 1 is more successful because it has strong binds where it needs to have and weak where it doesn't need to have good binds. So let's turn to how this research can apply beyond my paper. In regards to future work, experimental validation is a key aspect to improve the design system. An example of this is through surface plasmon resonance, which is a process that uses light reflection to analyze how well the aptamer binds. As you can see in the image, SPR will first shine a light onto an interaction and see the angle that it bounces back at. After an aptamer comes into the picture, if it binds successfully, there should be a change in the angle in which this light is reflected. This is a style of experimental validation and truly put my system to the test. We can also see an enhancement of the model by testing it in a real-world situation. For example, the solution around the brain, cerebrospinal fluid, is nearly 99% made from water. So the aptamer must be able to survive facing these environmental pressures. By enhancing the structure of the system, we can test it in a more real environment and ensure that it'll still be able to work. When we turn to the applications of this research paper, the impact is twofold. First, this system can target immunotherapy, expanding beyond cancer cells in the brain. Rather, the transportation can be adapted and altered with alternate components and pathways. With immunotherapy, this could involve targeting T cells to make the immune system stronger in response. And also, this method of transport system design can be applied to alternate cancers and vaccines, showing that there is a large scope for potential and it being adopted as an approach to cancer therapy. In conclusion, this research paper focused on designing an aptamer-based targeted drug system, specifically for glioblastoma cancer. Aptamers were essential to this process because they are easy to manipulate and their small size allows for efficient travel, especially through the brain. Through several means of computational analysis and simulations, Aptamer 1 is shown to be the best. My research lays the foundation for developing a more effective and less invasive glioblastoma treatment strategy, specifically through Aptamer 1's ability to cross to the blood-brain barrier by binding to the insulin receptor, then its ability to selectively target the cancer cells with limited damage to our healthy cells. Through future work and application, it's clear that aptamers are a promising alternative to antibodies, providing a specific, efficient, and targeted approach to therapy and delivery. Through greater research and investments, the possibilities are endless. Incorporating aspects such as fluorescent dyes or alternate drugs and interactions could revolutionize the way the medical world moves forward. These are my references, and thank you for your time.